Just hi everyone. We're just waiting for Jonathan to get the live feed going. Good.
It's going.
Good.
Give it like 15 seconds for folks to join.
Cool. Let's see how long I can stand here awkwardly, not saying anything. All right, good. Yeah, so just a few announcements. I'm Adam Ferguson, the collection manager of mammals, and I have the honor of introducing Dr. De La Sancha today. But before we begin, I just want to acknowledge that the Field Museum acknowledges that it resides within the traditional homelands of the Three Fires Confederacy, the Ojibwe, the Ottawa, and the Potawatomi. It recognizes and is grateful for the original peoples who laid the foundation for the city of Chicago and the diverse indigenous nations that reside here today. Just a couple of announcements too. After today's seminar, we ask that people please use the microphone when you're asking questions, because that way the people on Zoom can hear. We're going to start with questions from the floor first, and then Jonathan will help us with questions from the Zoom. And I also want to extend an invitation for folks to join us for lunch at the bistro after the talk. And with that, so it's a real pleasure to be able to introduce Dr. Noe De La Sancha for the Watson seminar speaker today. Dr. De La Sancha is an assistant professor at DePaul University, where he studies the impact of anthropogenic change on small mammal communities, including work on disease, ecology, and evolution. He has over 50 scientific publications in top journals such as Journal of Biogeography, Scientific Reports, and Ecology, a long history of obtaining funding, including funds for the National Science Foundation. He's also a former Ford and Fulbright Fellow. And you may know Noe, he's no stranger to many of us here. He's been at the museum for 11 years as a research assistant. As a research associate and in-house resident research associate. And he's contributed significantly to the collection of mammals here, both through active fieldwork and mentorship and training of students. So I first met Noe actually at Texas Tech when I started grad school, and he was kind of legendary by then already. He had spent so much time in Paraguay that people had rumors that he had bought a herd of cattle and was moving to Paraguay permanently to stay there. It turns out that wasn't true. He was actually just so busy trapping small mammals, building one of the largest data sets on small mammal communities in Paraguay's Atlantic forest, which is a project he still publishes on today. And in fact, he was just recently awarded the Biota Early Career Award from the Walder Foundation to resurrect that project in Paraguay. So Noe is here today to talk to us about mammals, museums, and models, methods to study the Anthropocene. And without further ado, Dr. De La Sancha.
Bueno, muy buenos días, damas y caballeros. Technically, I guess it's buenas tardes. Good afternoon, ladies and gentlemen. Thank you for being here. I genuinely appreciate it here. I was extremely excited when I was invited to give this talk, considering it only took me 11 years. So my talk is a little bit long. Next time, take a little bit less to invite me again, and I'll make it shorter. So technically, it's your fault, man. Just saying. Okay. So today I'm going to talk about sort of my research that I've been doing for a while. Big picture, I'm going to talk about three big picture projects. Some of it has to do with some of the research that I've been doing in Paraguay for a long time, a new project that we have from Ivory Coast from West Africa, and sort of ongoing urban ecology work that we started here in Chicago. So to start off, I am a new immigrant to this country. I was born in Toluca, Mexico. I immigrated to the States when I was really young. I grew up in the border. This has been my scene when I was growing up. There's nothing new. The benefit of growing up in the border in a low income, primarily Mexican-American community was that we had this next to us, which is a major landfill. When I was there, one of our high school teachers kind of got us to picket to make sure that we could get this thing out of our neighborhoods. And it got me thinking about environmental justice, but also environmental racism. As part of that, I ended up going to a really hippie school up in northern Wisconsin, about 8 hours north of here. If you think Chicago's cold, Chicago ain't got nothing in northern Wisconsin. And so after that, I ended up doing my PhD in the beautiful landscape of Lubbock, Texas. Yes. Okay. After that, I did a postdoc up in Rhode Island, and then I ended up teaching after I moved here with my wife to Chicago. And I started off at Chicago State University for about 7 years. Being a field associate since then, and now I just took a new position at DePaul University. All right, so why are we talking about this? So this sort of molded all my research, so my ideas of things that I'm interested in. First of all, in case you didn't know, and I'm sure everybody here already knows, and they should know by now, we're in the middle of the 6th mass extinction on our planet. There's been at least 5 major mass extinctions where anywhere from 70 to 90% of all biodiversity went extinct. And now we're right in the smack middle of the next one. This is the 6th one, which is the Anthropocene. For mammals, this is particularly important because especially in tropical regions of the world, this is where we have the most number of species. As part of that, the main reason why we're losing species is habitat loss or habitat change. As you can see, there's still many, many, many species of mammals that we still have not been able to document. And this is why museum specimens are so important, because this helps us to better categorize, quantify the number of species that we have, especially for ecological projects. Now, most of my research is here in the Atlantic forests of South America, which is where I'm going to be starting to talk. And as part of what we see regularly is this landscape now. Only about somewhere between 12 to 20% of the original forest is left. And when I was in Paraguay, as sort of commenting on what Adam was saying, when I came back, there was this rumor that I had started to wrestle cattle out in the middle of Paraguay. And in the 1990s, yes, cattle was a primary reason for deforestation. And so after a while, it started to become a little annoying to me, kind of a little disheartening, that people would think that I'd be out in the middle of Paraguay wrangling cattle, doing some kind of cowboy stuff. And it never occurred to me why people would think something like this. The interesting part was that I wish people would have given me a little bit of credit. Cattle was primarily the market in 1990. By the time I was there, it was primarily soy. So the market had clearly moved on. So that was a little frustrating that people wouldn't at least give me a little bit of credit. Since then, we started a new line. We're now in Trader Joe's. No, to be clear, soy is usually not used for soy milk. Soy is primarily used for cattle, primarily for feed. Unfortunately, in Paraguay, most of that soy is actually exported, so it's not even used by local farmers. It's usually exported to places like Asia, where it's usually used primarily for pork or some pig-related product. Once again, I was not involved in any of that. Anyways, so I primarily work on small mammals, primarily mice and marsupials in South America. The reason for this is they're small, they have high diversity. So anytime they're using ecological questions, they have high diversity, they have high reproductive rates. So you can go to a place, you collect a bunch of them, and you really are not going to impact their populations. They have limited dispersal. This means that whatever you catch there is probably what's living there. So they're good indicators, both for biogeographical questions, but also for ecological questions. Many of these are sensitive to deforestation, or they react quickly to deforestation. And many species in South America are actually endemic, especially for the Atlantic forest. Now, this required lots and lots and lots of years of initial work in the field. Usually field work is not easy. We were able to collect all the specimens when we were there. Here's a good picture. I like to highlight Lourdes Valdez right there. We'll talk about, just keep that there on the back burner for now. When you think about this, one of the things, I had all these ecological questions that I wanted to start tackling when I got to Paraguay. And one of the things that I realized very quickly is that we don't know squat about the species we have there. This is actually kind of cool. This was the first mammal paper I ever published. And I really appreciate it because it's actually included a first species record for Paraguay. And actually, as part of this, there's some, it included some specimens from the Field Museum, which were the first to be documented for Paraguay. As part of that, there's all kinds of subsequent notes, letters that had to do with taxonomy, with systematics of various species. We're trying to improve the understanding of what species we even have. As early as 2 years ago, we found a new genus record for Paraguay, and we're still trying to improve on that. This led to this checklist of mammals of Paraguay that got updated. And as early as 2 years ago, as I was saying, we've now added a few more species to Paraguay. There's 184 species, and we still have a lot to do. We still got a lot of basic work to do. So I really like this, figure. This is from a paper that we had a few years ago that sort of outlines all the big picture questions of all the different questions and aspects of things that I've been doing in my research. And as first of all, museum specimens are fundamental to all this work. But as you look at this, I would like to highlight sort of the different aspects of ecological questions that deal with populations, communities, ecosystems, landscapes, and then biosphere or biogeographical type questions, which we've been trying to address throughout the years. So to start off, most of our specimens are housed here at the Field Museum. As I was saying, this was extremely important just to get correct identifications of our specimens. And this required not only morphological, but also DNA work that we're still ongoing. And so if you look at this, the first thing that we want to look at is population level questions. First thing I want to highlight is this awesome paper by one of my first students at Chicago State, who was Gabriel Hernandez, who was working on population level analyses of morphology of Acheron montensis. And that was one of the main things that helped us to identify many of the specimens that were then important for subsequent work with ecology. Now, I want to point out this paper that was kind of cool. It doesn't have to do with the Atlantic forest, but it's a recent paper that we were able to outline that sort of shows how we're able to use morphological data to describe patterns at biogeographical scales. This was a cool paper that just came out. We were able to include various types of analyses, including regression tree analyses that allows to explain what is the variables that best explain morphology. So it turns out that if for this particular group that goes over the Andes, it was longitude, and we think we might have a new sort of description of a new ecogeographical rule for morphology that has to do with a rain shadow effect. Now, part of that, the next thing that we want to look at is community level questions. This was part of my dissertation work where we're trying to understand how the communities of small mammals throughout the Atlantic forest were shaped. Just this week, actually, we got this paper that got published in Diversity and Distributions, where we look at edge effects. And as part of this, we looked at multiple dimensions of biodiversity and what happens to small mammals as you get closer to forest edges. Now, one of the things you can consider is that we have more and more deforestation, that equals higher levels of edge effects. And so what happens here? This is part of the sampling that we did when I was in Paraguay and also subsequent trapping afterwards. We were able to look at various aspects and dimensions of biodiversity, including richness, which is the number of species, abundance, various aspects of functional diversity, various aspects of phylogenetic diversity, and also corrections for things like Shannon Diversity Index and Pailu evenness. One of the things that we found is that many of the aspects of biodiversity are orthogonal. That means that they are independent of each other. And this is one of the things to consider as we start thinking about how biodiversity changes, not only with deforestation, but with edge. Big picture, we were able to come up with interesting models via a mixed model approach that allowed us to come up with sort of regression style analyses that allows to include multiple explanatory variables, both quantitative, but also categorical. Big picture, this is what we found. We found that richness and functional richness were best described by generalists. So one of the things to keep in mind is generalists are taking over the world. And this is not only here, but also you'll see later in Ivory Coast, but also in urban environments. But not all generalists are the same. The first thing that we found is for richness, we saw very good evidence of a spillover effect. So richness was best described by species that were not from the Atlantic forest that were coming in. Contrary to that, once again, orthogonal to that, abundance, what we found was that abundance was best described by generalists, but these are Atlantic forest generalists. So a totally different ballpark there. Then things like Shannon diversity, Joe's corrected, Pilo, various aspects of functional diversity were best explained by this little generalist species, Ilamis megacephalus, which for the rest of its distribution is primarily found in cerrado habitats and little gallery forests in other habitats, and then they just kind of creep into forest remnants of Eastern Paraguay. And then phylogenetic diversity was best explained by the presence of this Atlantic forest marsupial. Okay, the next thing I want to talk about is this work by my first master's student, which is Damon Gonzalez, who spent lots and lots of time here. You might know him. And what he was looking at was the three most abundant rodents that we have in forest remnants of Eastern Paraguay. These are Acheron montensis, Acheron paranaensis, and Oligorhizmes nigripes. And one of the things that he was looking at was how do these species that are extremely abundant, how do they coexist? And so first of all, we tested all kinds of associations between abundance and the various species. Then we did some really cool assembly rule node models that allows us to assess what is the best sort of pattern of assemblage of these various creatures. Big picture, long story short, we came up with a resource partitioning hypothesis that best explains this scenario. The really cool thing about that is that we had really good ontogenetic series for each of these, and he was able to incorporate morphology on top of that presence absence null models. He was able to see how these things coexist, and we ended up using not only principal components analysis, but a phylo-PCA. A phylo-PCA basically extract all the signal of phylogeny and only leaves the ecological variation. Now, as part of that, he was able to do some really cool geometric morphometrics. And this is from another project that I was doing on the side a few years ago, where I was testing for bite force. And if you look at these three, these are Acheron montensis, panamensis, and Oligonigripes. They totally are not competing with each other. And we could see this from bite force because they're clearly using other resources. Once again, more evidence for this resource partitioning hypothesis. All right, moving along, landscapes. A big part of what I've been looking at is how deforestation affects Eastern Paraguay. We did this study in 2021 where we were able to compare forest remnants from 2000 and then from 2019. All the stuff in yellow is the stuff that was lost between 2000 and 2019. As part of that, we implemented graph theory to better understand how the whole network of the remaining forest remnants can be connected. Not only that, but we were able to see this based on dispersal abilities of rodents all the way to marsupials, to bats, medium-sized carnivores, and large predators. Obviously, this creates a difference in the matrices, and this allowed us to identify important remnants that should be protected for conservation value. Now, as part of that, we also did this on the opposite side of Paraguay, which is the dry Chaco. This is even more scary because in the Atlantic, in the Chaco of Western Paraguay, we're seeing a dramatic rate of deforestation. All of this yellow stuff is the rate of deforestation from 2000 to 2019. Once we quantified this, we ended up coming up with an area that is equivalent to the state of Indiana. Think about that. In 19 years, we lost an area the size of Indiana, gone. Now, this is a remarkable habitat that is very poorly understood. It's one of the few dry tropical forests on the planet. It's unique to anywhere on the world. We have a huge amount of unique armadillos that are not found in other places. We got primates, various types of other synarthrans, and even things like tapirs, giant anteaters, and jaguars. Many of these require this connectivity and these habitats. As part of this, once again, we were able to find important remnants that are important for conservation value, and we were able to show this. If you see these two, you can see how much of that deforestation has happened in Eastern Paraguay. And it's really scary to see what is going on here in Paraguay in particular. Paraguay, by the way, was the country that was most affected by the deforestation in the Chaco. Okay, moving on, zoonoses. Now, one of the things that we talk about is what is the impact, big picture, on populations and the species that are living there. This is a paper that we got in 2021, I think, where we were able to, using hair, we were able to sort of show that populations of small mammals that are found in smaller forest remnants are more stressed out than their counterparts in larger forest remnants. Now, the big picture is that if we have populations of individuals that are more stressed out in smaller forest remnants, that means that they're more susceptible to diseases. They're more susceptible to other types of transmission from one species to the other. And the other part of that is that as we have more and more deforestation, more and more people are living closer to these forest remnants. So the reason we're all here right now, why half of us are wearing masks, are for this exact same reason, right? And you know, we got lucky with COVID. There's a lot of other stuff out there that could really, really, really put a damper on things. So as part of this, I don't want to drag everybody down, but as part of this, we just got a Biota Award where we're going to try to start looking at the next level of stuff, where we go try to make the connection between deforestation, stress, and zoonoses to try to test the hypotheses that animals that are more stressed out are going to have more various types of parasites and pathogens in them than we would expect. So that's the big picture stuff of that. Next thing is going to be this biogeographical scale analysis that we did of small mammals along the entire Atlantic forest, from northeastern Brazil all the way to the Atlantic forest to Argentina and into Paraguay. The cool thing about this is this was a massive data set that we're able to get information about the species that were found in any particular forest remnant. We're able to get the size of the forest remnant when these things were captured, and then we're able to extrapolate bioclimate variables for each of these. And so from that, we were able to decouple or disentangle hypotheses about what causes taxonomic, functional, and phylogenetic diversity patterns. And we're able to test, well, is it deforestation that's causing this? Or is it some kind of historical bioclimate variable? Now, we found that this actually varies by groups. It's not that straightforward, but yes, we do lose a lot of species at the local level, but at a biogeographical scale, some sort of climatic variable is the one that best explains these patterns. Now, this might hint to you that if we have more and more climate change, many of these species assemblages are gonna start to change dramatically. So not only do we have to be worried about deforestation, but we really gotta be more mindful also of the impacts of future climate change. All right, that was a lot. Now we're coming back to the next thing that we started working in the last few years, which is our research in Ivory Coast. Now, the reason why we started looking at Ivory Coast, I'm sure everybody likes chocolate. How many of you like chocolate here? Oh, all right, there you go. Okay, it's not just me. Okay, so the thing about chocolate, right, is that most of the chocolate on the planet right now, anything that has cacao base, is produced in a tiny little country in West Africa called Ivory Coast, which is about the size of Montana. 40% of all cacao is produced there right now. Another 30% is produced neighboring country in Ghana. 70% of all cacao products on the planet come from West Africa, okay? Think about that. Now, cacao, for those of you who know, is originally from the Amazon. It's a tropical forest species. That means that you have to go and deforest a bunch of rainforest so you could grow cacao. Now, as part of that, in Ivory Coast, only about 10 to 15% of the original forest, the Ghanaian forest, is left. And as part of that, we started this project where we started to sample primary forests, secondary forests, rubber, fallow, and cacao farms to see the impact of cacao. And we use small mammals as a way to get at these types of questions. So we went and sampled various places. Now, this project, we're gonna start somewhere around, I think it was 2012, 2014. I was about to go, we were ready to go. And then there's all these Ebola outbreaks that started in West Africa. Once again, zoonoses is associated with deforestation and all these things that are happening. And then, so either way, we prevailed. We started doing trapping all over West Africa. In 2017, I was able to take some students of mine from Chicago State. We were able to do further trapping where we compared forest versus cacao farms. And one of the things that we were able to get was we were able to establish sort of baseline data for specimens from Ivory Coast, from West Africa, which are some of the least areas that we have in our collections here at the Field Museum. So, proving in that a little bit. As part of that, we were able to bring various students that helped us up with phylogenetic analyses. We were doing really good until the summer of 2019, and then everything stopped. And thank God, Molly McDonough came in and saved the day, and she's been actually helping us with a lot of the phylogenetic analyses moving forward. Who knew she was a Bears fan? And still, don't wait, a first round pick, I guess. Okay, anyways, as part of that in Ivory Coast, we were able to then start looking at this little mouse, which is called Praomys rostratus. And the reason for that is because once again, we came back to start like we did in Paraguay. We don't know, we haven't been able to identify all the species. We had to start using DNA markers to start looking at many of these. And as part of that, there was a awesome intern that I hosted here at the Field Museum, Rachel Kreitz, who started looking at variation of these. At first, we thought that there was two species, one species that was found in modified habitats and one in the forest, the Ghanaian forest. Now, it turns out after some DNA markers is that they're all the same species. So we're like, what? How are they partitioning here? And we started looking into implementing geometric morphometrics. We found that there's no evidence of sexual dimorphism. So it wasn't any kind of sex that was creating our clear differences in our specimens. Once again, we're very fortunate that we had entire ontogenetic series of these. So we're able to compare from really young individuals to really old individuals and see how they compare. We tried many ways to understand how these things are partitioning. And then it occurred to us, why don't we start partitioning them by habitat? And sure enough, the thing that we found is those are all significantly different. My point is that as we start modifying habitats, we start creating microclimates. We're also creating microevolutionary processes that are changing these species already. And we see this just from these specimens that we found. All right, moving on to communities. Big picture, we're starting to figure out what is happening with the forest versus cacao farms. If you can see right here, you can see the cacaos here in blue. I think it's blue. I'm partially colorblind, so I'm assuming it's blue. So cacao, you see the cacao farms right here, and you can see primary forest over here. This is Jacquard diversity indices, which is highly biased by richness. Bray Curtis is highly biased by abundance. Remember I said that those are orthogonal to each other, right? So as we see this, we can see that cacao, or rather the assemblages that are found in cacao farms are very, very, very different from primary and secondary forests. So clearly this thing is having an impact on those species there already. We want to move on so we can start looking at various types of diversity metrics moving forward, and hopefully in the next few years, we can start getting some of those papers out. Now, as part of this, the one thing that was missing in this whole figure was this whole thing right here, which is urban ecology. If we're going to talk about the ultimate land modification, it's cities. And then it occurred to us that we have all these cities right here, so we can start doing some cool ecology, urban ecology work. So what we started here with Felix was this whole idea. We started thinking about this idea of population dynamics and morphometrics of urban brown rats, which is Rattus norvegicus in the Chicagoland area. Back in days when we had happy hours, many of us would meet with each other, and we started philosophizing about how can we test rats. It turns out that supposedly rats in Chicago, we are the rattiest city in the US, or so they claim. And there's lots of reports that supposedly back this up. And we thought, if only we could test this through genomics. And then this paper came up from New York City, so they're beating us. As part of that, we actually started a collaboration with the Lincoln Park Zoo, and we sort of got squeezed into this NSF grant where they were looking at various pathogens and also people's attitudes towards wildlife in urban areas as a function of involvement with rats. Now, they're doing some really cool stuff, and we were like, okay, let's do this. We got all these rats from the Chicago, from the Field Museum, but what happened was that we ended up with only 40 specimens from Illinois, 30 of which were from Cook County, and only 14 of them were from the Field Museum alone. So basically we had no rats to compare. So since we started this collaboration with the Lincoln Park Zoo, we've acquired 123 rats that now we've sequenced, and we're processing those as we speak. I think next week, we're processing another 150 rats. All of this is going to be used for both genomic and morphometric analyses. As part of that, from the genomics perspective, these are some of the questions we want to do. Is we want to determine the origin of city rats here in Chicago. We want to calculate population genetic variation and see, we want to test to see if Chicago rats really are the rattiest in all of the United States. Could be. As part of that, we had an awesome intern that was helping us with the initial sampling of these specimens. These were sent out to Wisconsin. We're cranking in 2019. And again, once again, COVID hit us. Since then, we've actually been able to get genomic data. And once again, as I pointed out, here is Lourdes Valdez. Lourdes was actually back with me in Paraguay back in 2008, way back in the day, back when we were young. And since then, she went out to do a master's in Chile. She got her PhD in Chile. She's killing it. And now she's here helping us analyze our genomic data for rats. Now, we were able to do RAD-seq sequencing. We're still sort of preliminary data. We're trying to figure out what we have. We have evidence that there's pretty good structure in the populations of Chicago. We're still trying to figure out exactly what that means. As we speak, she's compiled another set of approximately 800 Rattus norvegicus from throughout the world. And we're gonna try to come up with a big sort of global phylogeny using genomics. And as part of that, the next part of that is the morphometric component. Once again, this paper came out from New York. So close. They're just right ahead of us. And they were able to see some cool analyses. And as part of that, we've been actually been able to clean our specimens. We're trying to identify and quantify functional traits. And as part of that, we want to get at morphometric questions dealing with rats of Chicago. This last summer, I had 2 interns that actually started to quantify and do geometric morphometrics of those rats. As part of that, we've now then coupled not only rats, but now we started to see, we wanted to compare not only the invasive species, but now the native species, which are gray squirrels, to sort of see if there's complementary of those patterns, not only of genomics, but also of morphometrics. Here's a little gray squirrel. They're so cute. This last Christmas 2021, I think, we had this awesome effort by all kinds of people, all mammals or mammalogists came out and started helping us process some of those. And we were able to get, we have tons of skulls now for morphometrics. And we've already processed the first genomic samples that we now have. Once again, the really cool part about this is on top of that, the Lincoln Park Zoo is actually analyzing all these other things associated with the rats. They're looking at lepto, hantavirus. They're looking at RSV, COVID, influenza, monkeypox, rodenticide, and they're doing isotope analyses. They're actually collecting ectoparasites and endoparasites. So this is all data that's going to be associated to our field museum specimens. That's an amazing resource, not only for now, but moving forward, which is pretty awesome. From there, then we started to expand with Lincoln Park Zoo, and now we're starting to look at how rodenticide moves up the food chain. And as part of that, we were able to get raccoons, some possums, and some skunks. Moving on up. Okay, finally, one of the last projects that I have is this NSF grant that we got awarded in 2019, which is digitizing, the acronym is FUNKY, and it's Functional Quantitative Characters for Ecology and Evolution. And as part of this, we want to actually create 3D models of approximately 1,700 rodent species from throughout the rodent tree of life. Now, in case there's anybody who's wondering, if there's any doubt that Stephanie Smith is one of the coolest people here, back when we were in COVID and we were all on Zoom, she actually literally sat with me on Zoom and we actually built this gaming computer. And look at that beauty. So as part of that, now Stephanie was also extremely generous and helped us with some training of some students. We're now, so as part of that, so this is part of our funky thing. And one of the things, for those of you who don't know, Overt is a repository for basically all the files for creating from CT scans of various specimens. Overt is this project that's trying to target all kinds of vertebrate species from throughout the vertebrate tree of life. As part of the funky, we sort of specialize on the rodent part. You can see in blue, these are all the CT scans that we should have access to now that we have processed. We're in the second round of that, and we're gonna start CT scanning more rodents in Michigan. If you see this from the Field Museum, one of the things that we're missing is all those specimens over here, which are primarily African species. So that's what we want to target next. We got quite a few. We got about 25 families, 128 genera, 264 species. Anybody wants to throw in some more rodents from there, maybe from like the Philippines or from other parts of Africa or other places that we haven't targeted, that'd be awesome. This is my new student, Lisette, who really likes to get her hands dirty. And she's been actually helping with this, and she started processing this, and this is our specimen she sent me yesterday. So we are cranking those out now, and hopefully we'll start having like 3D models. This is a PDF, so I can't show you how it rotates and does some really cool stuff. Okay, so as we're closing off, this is one of our pictures from one of our last dissecting of Meso carnivores. And I really like this picture. And it's for one very specific reason. If you look at this, everybody in this picture represents somebody who is not represented in STEM and in science. Everybody, okay? No offense, there's no white guys, right? And that's one of the things to consider. And that's one of the things that we got to consider moving forward, especially in science. And also, when we think about natural history collections and all this stuff, people of color are not just gonna come here, okay? We have to make an active effort to recruit them and to keep them here and keep them happy. You know, this isn't gonna happen just because we're so cool. Okay. If you think about it, especially working at Chicago State for so long, one of the things to consider is that many of the people who come from underrepresented school were people who come from underprivileged backgrounds. Many of these people are the first in their family, like I was, the first one in my family to go to college, the first one to actually go get a PhD. I was the first one to, when I told my parents that I was gonna go study rats, they were like, are you kidding me? Like, I thought you were gonna become a doctor or veterinarian or something, or you're gonna make money. That's the point, right? We can't just assume that we're so cool that everybody's just gonna come here, okay? And this is why I really value this thing. And one of the things that I found, and actually I legitimately Googled this, where if you Google this, look up white men white men are judged on their future potential. And I looked it up and literally all this stuff came up, not only from news articles, but also from literature, from academic literature. White men, think about this, white men get judged on their future potential, whereas women and people of color get judged on what they've accomplished right now, okay? It's true. You see this in business, you see this in economics, you see this in STEM, you see this in all museum-oriented programs and whatnot. And so the point to be made is that this is part of everything moving forward. It's complex, it's not easy. If we're gonna improve science moving forward, we have to incorporate more people. Ideas come from all kinds of places, right? Not just from the same phenotype. So that's something that we have to be very mindful of. So big picture, this is where we are. We're just the tip of the iceberg. We have a lot of work to do, lots of things to get done. I got lots and lots and lots of people who I need to thank. Many of my students at Chicago State. And more than anything, I need to thank my awesome family, my wife and my 2 kids. Yeah, there's been lots and lots and lots of awesome mammalogists that have come through here. Anderson Feijó, brilliant mammologist. Daniela Rossoni, another brilliant mammologist. Renan Maestri, another brilliant mammologist. Julio Vilela, Patti Perez, Natalia Cortez, many great mammalogists that I come through here that I've been able to share some space with. Many of my collaborators. I also want to thank everybody at the Field Museum who's always been extremely helpful with me. I wouldn't be here without the support of Bruce Patterson, so thank you all. Now, if there's any questions, you can ask Adam. I gotta go. Yes, ma'am.
I have 2 questions. One is like not really a question, but you mentioned South American marsupials. I'm not sure people know what those are. So like what kind of marsupials are in South America, basically?
That's an awesome question. What are the marsupials in South America? Well, it turns out that in the US and Canada, we only have 1 species, right? Which is the Virginia possum. If you go down into Mexico, we get about 12 now. And then once you start getting to Central America, that increases. By the time you get into South America, there's over 100 species of marsupials, okay? Many of these look like possums, sometimes of all kinds of sizes. So some are really large. They kind of look like our possum. Some look like smaller versions of those. There's some that are arboreal, some that have little short tails, which are little things like monodelphis. There's also things like water possums, which are beautiful creatures. They have these little webbed feet and everything. And interestingly enough, that's interesting you mentioned that because all of the, and you look at the fossil record, there was everything from large saber-toothed possums running around to like these other herbivore, 4-legged kangaroo-looking things, right? And if you think about it, well, if you start thinking about the evolution of marsupials, the oldest fossil record that we know of from of marsupials comes from somewhere in China. And then the second oldest was from Canada for a long time. Then somewhere around 65 million years ago, 70, they came into South America. They became highly radiated. And then through Antarctica, they immigrated into Australia. So yeah, to answer your question.
So the other question is, what do you think will happen with squirrels versus rats?
What was that?
What do you think will happen with squirrels versus rats?
What do I have it?
What do you think will happen?
Oh, what happened? Well, I don't know. That's a great question. We're still processing the data. We literally were able to process specimens last Christmas for the skull. Right now, we still have genomic data. We only got like a third of our genomic data. We still got a long ways to go. So there's a lot of potential there for the next few years to try to process that data. That's why we recruited Lourdes, so we can try to start getting at those questions. So in 11 years, when I get invited again, then I'll answer that question.
Okay.
Julian.
Anyway, thanks a lot, man. Still trying to catch my breath from everything you went through. But I got two questions. First is kind of trivial. Second, a little more thought-provoking. First one is, you talked about, you talked about bite force as a way to discriminate sympatric species in Paraguay, I think.
Yeah.
How did you measure bite force, and why not just look at like size of the sagittal crest or the breadth of the zygoma?
That is a brilliant question. First of all, for lots of them, they don't have sagittal crest, for starters. The next thing to think about is like there are multiple ways of quantifying bite force for rodents. Is really cool because if you look at the lower mandible, there's a little formula that was created where if you measure the incisors on rodents, and if you get a longitudinal measurement versus the width of that incisor, there's a little formula that correlates very highly with actual bite force from a little instrument that people use to measure it from animals biting on it. That only works for rodents, which is why it was limited, and so our bite force was only for rodents. With various other mammals, there's really cool measurements that you can take on the mandible, like 3 or 4 major measurements. There's a formula also that allows you to quantify bite force that's highly correlated with actual measurements from real experiments. I can send you that if you like.
Yeah, so you did have morphological.
Yes, 100%.
Equivalence. And then the other question is, when I was a kid growing up in Chicago, we had 2 species of squirrels, Niger and Carolina gray squirrel. And so Niger, as far as I know, is extinct in the city. Maybe it's around in the forest preserves. But that kind of competition back then, Carolina squirrel won. And might there be anything interesting to look into regarding previous competition, how that might have impacted morphology, and now there might be a release because one is gone? Or can you measure stress still in dry museum skins where the Carolina squirrel might have been stressed and now it's not since it's king of the roost? But just speculative food for thought.
Those are all awesome questions. Yes, so right now, technically in the Chicagoland area, there are still 2 squirrels at least. Gray squirrels are the most predominant ones. There's a lot of really cool work by, I think it was Joel Brown at UIC, where he was looking at how both of the squirrels are separated. If I remember correctly, there's something along the lines that some species are highly predominant in highly affluent neighborhoods, and the other species was primarily found in low-income neighborhoods. I forget which is which, but I can look that up. I can certainly send you that literature. But the thing is that they're partitioning space, and it probably has to do with resources. Yeah, but that's a great question. Yeah, stress. We look at stress mainly on things that were a little bit more recent, like the stress analyses that we did were from specimens from about 2008. It'd be interesting to start looking how stress or that hormone still persists in the hair after like 10 years or something. That'd be pretty cool. We have not been able to do that. Up to very recently, we didn't have the money to do that, but maybe now we can start looking at some of those things. But those are awesome, awesome points. Thank you. Yes, ma'am.
Really nice talk. No, I really enjoyed that.
Thank you, ma'am.
And am amazed by the sweep of your research program.
Thank you.
My question is more about one of the settings of your studies rather than the studies themselves. In the cacao plantations in West Africa, my understanding was that cacao does better as an understory tree. And so it seems kind of counterproductive to clear rainforest and then plant cacao without cover plants, unless they're not completely clearing.
So the question was along the questions of like cacao doing better in understory. It actually, that's, okay, so here's the thing. For commercial chocolate, in order to get the amount, the actual quantity of production and at the speed that you needed to keep up with the commercial market, it's all sun grown. And if you look at most of the almost entirely, all of the production in Ivory Coast, it's all sun grown. They don't have shade grown coffee, shade grown cacao or coffee for that matter. And if you look at it in Ghana, research from Ghana and from other parts of West Africa, there's been plots of doing this. But a lot of that shade grown cacao is more for like a boutique or for very specific, like shade grown cacao products and stuff like that. And it's actually a tiny fraction of the overall market. And it's interesting that now 20 to 25% of the rest of the cacao that's not grown in West Africa is grown in Indonesia. And that's the next frontier where they're just clearing the heck out of everything to keep growing cacao. The interesting part is gonna see what happens with climate change, because as those climates shift, you're gonna have to go to higher latitudes or higher elevation. The thing about cacao is that cacao takes somewhere between 7 to 10 years before we start getting product. We actually have like full pods. So this isn't something that can just shift overnight. You know, this isn't like a product that you could just go start growing somewhere else. It's gonna start growing, it's gonna be fine, especially at the commercial grade that we have it now. I would say enjoy your chocolate now as much as you can, because it's gonna become more expensive, and it's definitely gonna become harder to attain for sure. But yeah, sorry. I'm gonna bring it right down. Not cacao, not chocolate. Yes, sir.
Hi, fantastic talk. I was just wondering if you could touch a little bit on the viral pathogen stuff you were doing. For the Chicago data set, and I think you're doing some other stuff too. I was wondering, so you're doing mostly like genomics level stuff, like metagenomics, or is it more like PCR and like seroplasticity stuff? I was wondering.
That is a great question. So you have other questions? Sorry, didn't mean to cut you up.
Oh no, that's it.
So the viral stuff, a lot of the viral stuff that we've been doing is basically led by the Lincoln Park Zoo, and they're sending this off to laboratories to do the processing. I think some of it has to do with just presence, absence of various pathogens, and some of them they're actually doing screening via PCR and some kind of sequencing of some kind. I'm assuming it's some kind of short thing that's specific to some of these viruses. Because the idea right there is just to get presence, absence of many of these, right? So I don't think they're going to use any of that data for any phylogenies or anything. It's just, do we have it or not? In Paraguay, we're trying to look at different alternatives. We haven't quite figured that out. This last December was my first trip in almost 6 years that I went back to Paraguay. We actually went to find actual sites. We're going to try to do probably some sort of mark recapture grids. We're hoping that we can implement some kind of nanopore technology so we can actually start screening those efficiently and quickly. In March, I'm going to go back and we're actually going to start testing that. So not quite there yet. That's our hypothesis right now. We can do it through screening. Yeah, that's where we're at right now. Thanks for that.
Great talk, Noah. Loved every second of it. As someone who has worked with your students before, and I have loved working with your students, I was just wondering if you could touch on your strategy for mentorship and training.
It's a great question. Mentorship, oh man. Okay, so yeah, so it's interesting because one of the things is that you gotta, well, there's several things. First of all, when you're trying to bring in people, you got to actively, lots of times you got to actively go and recruit them, right? And you got to try to get them to come in. I think for a lot of these disciplines, unless there's somebody who is not already predisposed to this type of research, you really got to, give them something worth doing, At Chicago State, one of the things is that we had to find mechanisms to pay them so that they would come here. Many of our students, especially from underrepresented backgrounds, they can't afford to come here to do this for free, just as a internship type thing. We need to find funds to recruit some of these students. And then the interesting part is coming up with mechanisms to have them stay. A good example is my master's student. Right before he was even before he finished his master's degree, he got a interesting job at a chemistry related company. And before he had even finished his master's, I think he was making more money than I was at that time. And so that's one of the things you can consider, especially when you have students that come from non-traditional backgrounds. Now, the other thing is that, it's complicated, right? Because for me, it's important to give people freedom to do whatever they want to do their thing. And the other thing to keep in mind is that you have to empower people to take ownership of that project. Sometimes it works, sometimes it doesn't work. And sometimes you spend a lot of money and get nothing in return, which is part of it, right? You just got to try to invest in people, and hopefully they will counter that. And more than anything, try to, as much as possible, do positive reinforcement, like more of a carrot than a stick, I think. So, and I don't know, whatever that's for. You had a question?
I've got a more general question.
Yes.
And that is, deforestation is the main cause. Legitimate deforestation, meaning farmers have the right to do that, versus private corruption, that they're getting something out of it. Where is the major problem, and what could be done about slowing deforestation?
And that, my friend, is $1 million question. So yes, you're right. At many scales, small farmers deforesting a little, 2-acre plot, that's not the issue, right? That's not the problem in the grand scheme of things. Big pictures, when you have things like this, like in Paraguay, you would have, 100,000 acres of just soy, like a sea of soy. You see this all over Paraguay, you see this in Brazil. All over Brazil, especially in Brazil, it's starting to go all over into habitats it's never been before. It's starting to go into the Pantanal. It's starting to go into the Amazon. It's starting to go to stuff. Most of that is from large-scale corporate soy production. This is the same thing with palm oil in East Africa, right? A lot of this are these massive, huge farms that are owned by, corporate entities and whatnot. It's a large scale of this. Having a little small plot where you grow your corn and some beans and stuff, that's not the main issue for deforestation. It's a large-scale corporate the agro-business complex, right? That's what it is, right? And you see this a lot. Interestingly, this was really sad to see. This last time when I went to Paraguay, apparently they're creating this massive trans-country road that's gonna go from Chile all across the Andes, through northern Argentina, through the Chaco of Paraguay, all the way to Brazil. It's gonna create all kinds of opportunities for people and stuff. But the biggest opportunities that it's gonna produce is for deforestation. In that Chaco paper that we're looking at, the reason why there had not been deforestation in the Chaco was because there wasn't any arid resistant soy. The second that came into the market, boom, started deforesting like crazy. I have lots of friends who are from the Chaco. They would tell me that they saw themselves where they would set up these bulldozers, right next to each other for about, I don't know, half a football field or something with massive chains and literally just go down, just start tearing stuff out, So the scale of that is more industrial now. I mean, someone with a machete isn't doing this. Well, I mean, they probably are a little bit, but that's not the big problem. The problem is those bulldozers that are literally coming in and dropping everything.
What could influence the corporations to change?
That's a great question. So That's actually a great question. My wife, for a long time, worked with Mars Company, which is the main company that deals with cacao deforestation. They've made a pledge that they're going to have deforestation. Actually, I probably shouldn't be talking about this. Anyways, so they've made some plans, some commitments to stop deforestation. Their way of doing this is still unclear. They just made this announcement that they're going to do this. Many other companies have done this, but most companies still don't have a plan. And this is where it would be very awesome if they start hiring mammalogists or ornithologists or whomever to actually start helping them on better executing those plans. But unfortunately, that, to me, that corporate science link is still not there yet. That's a great observation. So.
First of all, Noé, thank you so much for your presentation. It was amazing. Both the science part and the social part of the issue you told, because several times we don't see this kind of discussion. We really need. But I have a question specifically about that study about the stress, the physiological measurement of stress in mammals in Paraguay. Did you have some morphological thing done with that material? I mean, because we work a lot with fluctuating asymmetry as a proxy of stress, but we are frequently assuming this stress because of the environmental condition, and you actually have the measurement of stress. Did you try to do something like this? Assess the level, the difference in levels of fluctuating asymmetry and comparing it with the physiological measurements you have?
That's a great question. We've not done that yet. And actually, way back in the day, when I was first starting, that was one of the things that I wanted to look at was fluctuating asymmetry, to start comparing one side to the next. For a long time, one of the issues that you point out, I think, is that many of the species that are that stressed out that you would actually see physical deformities tend to die and we never catch them, right? And that was always a big sort of problem that we had with some of those things. We've been looking at some of these possums, and if you're interested in this, you should check them out. Some of the possums from Chicago, we had one where the snout was completely to the side.
Fossils are kind of.
Yeah, I can definitely start showing you some of those things. So it is potentially real, and it'd be interesting to see if we start looking at this with association to actual stress hormones, right? I've not had the time to do that, but you know, anybody wants to collaborate. If only I could find a young whippersnapper who could help me collaborate on that. That would
we
be
should
awesome.
definitely talk about it.
We'll definitely talk about that. I'd be happy to talk to you about it for sure. But the specimens are upstairs.
Nice.
Any other questions? No more questions? One more question. There we go.
What trap did you use to get all these species?
That's a great question. So for the Paraguay stuff, we actually use 3 types of traps. The traditional Sherman live trap, which is this little trap that's made of aluminum. It has a trigger mechanisms. The animals go in and it basically captures them alive. We also use the traditional snap traps, which is what you would put in your house and it actually gets a specimen, you know. And then the other thing is we actually use pitfall traps. And pitfall traps, if you're not familiar with it, you have something like a regular paint bucket, right? And basically you bury it in the ground at level of the soil. And then what we did is we put what's called a drift fence, which is this massive sort of barrier. And the idea is that the animals come, they hit the fence, they go and then they fall into the bucket. With those, sometimes you got to put a little bit of water to get things from jumping out. This is one of the to-do lists. We want to quantify the different trap success between each of those types of trapping. The interesting part is that for each one of them, we got different stuff. Not only different species, but different demographic groups. With the pitfalls, they were extremely valuable for juveniles that you just never caught in those other traps because I think they were too small and they never got the mechanism to actually close. But with the pitfalls, we were able to get a bunch of juveniles. That's how we were able to do a bunch of the allometric series stuff, is from a lot of the pitfall stuff that we actually captured. Yeah, those were the three main mechanisms. Oh, and then with this new round of stuff, we started to implement camera traps. I didn't have time to even talk about those camera traps. But camera traps, they're these little cameras that have some sort of trigger mechanism for movement, and then you can get pretty awesome videos. If you look at my website, we have some from Ivory Coast that we've caught before. Yeah, we're getting some cool stuff. Other questions? Comments? Answers? Hypotheses?
Sorry, I have my watch. I'm not keeping time. Yeah, thanks, Noah. Thanks again for your good talk.
Thank you very much. Appreciate it.